cancer cells behave and act a lot like neurons. And I think that was a big surprise for the entire field.
We're in the game of actual human beings who are dying quickly, and we're trying to get technologies that can help those patients as quick as possible.
Today, I'm going to be talking to Ben Woodington and Elise Jenkins, who are the co-founders of Coherence Neuro, a startup that is building therapeutic neurotechnology that manages cancer from inside the body. I first want to talk about what specific device they were building, because I think it really sets the stage for how interesting the Coherence pitch is. Ben and Elise, welcome to the podcast. Your first device is called SOMA. What exactly does it do?
That's a brilliant opening question. Thank you so much for having us here. To rewind slightly, so you're right. We build technologies that interface with cancer and surrounding biology of cancer using electrical stimulation and recording. And we're really leveraging this intrinsic electrical properties of cancer, and also the way that they intersect and interact with our nervous system. We have a lot of programs ongoing, and I'm sure we'll talk about some of them today. But as you correctly identified, our first product and program is SOMA, which we're using in brain cancers. And this is a tiny device, a BCI-like device that sits in the skull, and it can deliver an electrical stimulus to tumor in the brain. It can also record the electrical activity from the tumor and around the tumor for readouts. And we're working very hard to investigate what those electrical readouts can mean for diagnosis of the patient, for the prognostication of the patient, and of course the therapeutic potential of that device as well.
Mm-hmm. The interesting thing that when I was first researching Coherence is that this is not the first device that uses some notion of electrical fields to interact with cancer. There's another one called Optune, developed by a company called Novocure. Is SOMA fundamentally different from the technology employed there?
Fundamentally different, yes. I think the natural development of, we have often said that Novocure is a 25-year-old technology, Optune is a 25-year-old technology, and if Optune is the Walkman, we're going to be the iPod. It's a lot more technically dense. We're doing readout capabilities, recording capabilities. The thing is much, much smaller. We get much closer and in contact with the tumor and the body itself.
And so maybe just to give some context as to what the Novocure device actually is, my understanding is that it is a non-invasive device planted, like you stick it to the side of your skull, meant for glioblastoma patients, emits a low frequency electrical field, preventing, fast dividing cells from dividing. What is the evolution of SOMA? What about SOMA is an evolution from that?
I'll let Elise clarify the Novocure point, because that was the background of her PhD.
Okay.
Yeah. So Novocure, as you said, it's a wearable device. It's actually 4 patches or arrays of electrodes that are positioned on the scalp, a shaved head. And it delivers alternating current electric fields. So they're actually high frequency, around more intermediate frequency electric fields. And they are proposed to interfere with mitotic spindle formation, the way that cells will divide. That's what Novocure's technology does.
And what about SOMA is what is the SOMA device an improvement on?
Though the Novocure device is powerful overall survival statistics, there's a very steep usage effect curve. So when patients are using the device sort of median time, which is around 18 hours, the overall survival in those glioblastoma patients is 4-ish months. But when they are super users of that device, so that when they use it 22 hours, 23 hours, or even more, that overall survival goes through the roof. And those patients are getting maybe 9 months, maybe more, median overall survival, which is almost doubling their life expectancy, which is huge. That's probably the most impactful thing in glioblastoma, in the last 20 or 30 years. Now, it is a very natural progression to say, okay, why aren't patients using that device 22, 23 hours a day? There's a large compliance issue. And go inside, and you're in charge then of how much stimulus the patient is getting and for how many hours of the day, and you offset a lot of those systemic effects. Why the patient wouldn't be using it that many hours a day, the skin irritation, just the fact that they have to wear something on the head all the time. And patients tend to not like wearing large things on their head. That's why there's been so many EEG device failures. Yeah. So just being able to justify going inside and treating those patients 24 hours a day is a huge benefit already, before you even start talking about the data elements and the recording elements that you can introduce, or the novel stimulation regimes that you can start using once you're inside.
What other electrical things can you take advantage of when you're actually physically inside the body?
I think there's a few things. So if you think about the way that an electric field is delivered through Novocure's platform, they require a very, very large sort of voltage to overcome the skull. So there's a big loss component there. And because of that, they have to carry this very large backpack, carrying a big battery pack, like a car battery, in order to deliver the electric field threshold that they need to enable that kind of interaction that they have with the cell, whether that be mitotic spindle formation or one of the other mechanisms, which is around a process called dielectrophoresis that happens in the cell during the metaphase. So there's like two sort of interactions that happen when you expose that kind of electric field to cancer cells, and they do that externally, using this field. The advantage of going, into the brain is that you no longer have that barrier anymore. You don't need these extremely large electric fields. You don't need these car batteries. You can have a very small wearable that interfaces. It doesn't, create. It's like non-obtrusive for patients. Like, there's a big argument around non-invasive versus non-obtrusive, and so that's one of the natural progressions in terms of using this type of technology continuously, which is shown to have the biggest benefit in patients. Is they're going in already. They're doing surgery already on these patients. Let's go and put a device in that can deliver that kind of field or other type of electrical stimulation. Let's do it locally. Let's also record what's happening, because we're right there. We're interfacing with that cells or those tissues, and we can do it without being obtrusive to patients' life.
And the actual device itself is emitting the exact same type of field that the Novocure device is emitting, or something else?
It can do, we can do either. So we've been looking at, again, like a lot of my PhD work was looking at Novocure's types of stimulation, tumor treating fields. But the advantage of being closer is that you can start to look at different types of stimulation. So neuromodulation is something that we are particularly interested in. And we've been exploring different ways of optimizing electrical stimulation in these types of cancers. So neuromodulation is a really interesting one. When we started development of the SOMA platform, we were really interested in the data that you could actually record, and that kind of was. We knew that there was a therapeutic intervention that you could use. Novocure had already shown that clinically. We were really interested in the data element. When we started looking at what happens longitudinally when you record electrical activity from these tumors, based on a lot of history of neural interactions that happen with these cancer cells, we kind of started to discover that there was these very interesting biomarkers that are really relevant to what people target with neuromodulation. And that was what drove us to consider, beyond just what Novocure are doing with electrical stimulation, field-based, mitotic spindle sort of cancer cell focus, to, well, what's happening in the rest of the environment, and how can we actually target or look at the rest of the environment, modulate that behavior, and how will that affect cancer cells? And so that's what we've been looking at optimized strategies for stimulation there.
Just to have a good mental model for what the SOMA device actually is, and where it is placed in relation to the cancer itself. Is it should I imagine it as you have a little head right here and a bunch of spikes coming out of it that poke directly into the cancer?
We wouldn't use the word spikes.
Okay, yeah.
We would use leads or threads.
Yes.
But yes, the brains of the device is that part that you see that's anchored in the skull, and as Elise said, that's delivered at the time of surgery through a very, very small perforation in the skull. Then the front end of the device is modular. So we have leads and threads that come off the front of that device, and we can position them in and around a resection cavity after a tumor's been removed, or into a tumor maybe without resection. Then in the rest of the body, we can look at targeting specific nerves or tumors there as well. And as Elise kind of alluded to, we've seen some pretty promising results there, looking at actually neuromodulation, so slightly lower frequency regimes rather than super high frequency regimes in those peripheral cancer indications as well.
Whenever I've brought up coherence to other people and mentioned neuromodulation in combination with cancer, there's always this surprise that neuromodulation does anything to cancer. What is intuition for why you would expect neuromodulation to do anything to the cancer? I can buy that monitoring the nervous system nearby the cancer helps you have some notion of biomarker, but why is performing neuromodulation at all affected?
So I think maybe the same surprise that others might have when they first hear this is kind of the same surprise that people who discovered these interactions had, that similar surprise, is that cancer cells behave and act a lot like neurons. And I think that was a big surprise for the entire field that made these discoveries. Mm-hmm. So a lot of cancer cells, and not just in the brain, this happens in other organs as well, a lot of them mimic a lot of the behavior that neurons have. So they hijack neural pathways, they have an ability to attract neurons into their environment. They also have an ability to track nerves into their environment. So if you're outside of the brain, and all of these properties make a really nice opportunity for you to then consider neuromodulation or other target strategies that are not just looking at the cancer cell itself. And you could consider similar analogies with the immune system, right? We're not when people are targeting, in some ways, when people are looking at immunotherapy, they're not targeting the cancer cell. They're targeting a completely different subsystem in biology that they can leverage and tune in a way to target cancer cells. And when people have started to unpack this opportunity that cancer cells are behaving so similarly to neurons, then it kind of massively opens up the therapeutic opportunities that you can exploit that we have used for decades in other indications. And so I think that similar surprise was also a surprise to the people who discovered it.
And that's recent. As well.
Recent.
These are recent discoveries over the last 5-10 years and today, not sort of mechanisms that have been uncovered, discovered, explained for 50 years or so, which is the exciting thing.
Is it fair to say that the glioblastomas have the most nervous system interaction, and maybe prostate cancer has the least, or is it not as clean as that?
I can say one thing, and then maybe Elise can say the other. It's difficult to draw a side-by-side comparison exactly between cancers and peripheral cancers in the body, simply because of the nature of those tumors. So the tumors in the brain are in a sea of neurons. It's a volume of conductive tissue, neural tissue, whereas tumors in the rest of the body are heavily innervated with nerves, but are not sort of existing within this sea of neurons, right? Yeah. So it is it's tricky to draw exactly a side-by-side comparison. It does change how we design the devices and how we introduce them to the body. But yes, they do still have these neural features. Probably you'd like to expand on the neural involvement in both. Yeah,
I think that, if let's say you were studying gliomas or tumors of the brain, I think it's an obvious, natural, I guess, curiosity to imagine that cancer cells might have some type of similar features because they're an extension of oligodendrocytes or, astrocytes or whatever other brain type glial cell. So it's not so unbelievable to think that cancer cells might behave similar to the environment that they're in the brain. And I think that's why a lot of the research has been more well established in the brain. So in gliomas and diffuse, in DIPG, which is a pediatric glioma, a lot of the research is very well established in those regions. But I also think that that's just a consequence of proximity, so how close they are to that environment. When you start to look at these interactions happening in other organs, I don't think it's a matter of proximity. I think it's just a consequence of the fact that people have done a lot of research already in glioma, and now that people are starting to observe these interactions happening in other organs, it's exploding. And if you were to do a PubMed search on cancer neuroscience and look at the trajectory of publications that are coming out in this space, it's exponential. And I think we're only at the very, very start of that now. So I think we will start to see that these interactions are possibly happening in every cancer in the body, not just ones in the brain. You mentioned that a lot of this research is relatively new, past 5-10 years. How was it first established that there is any crosstalk between the cancer and the nervous system?
By one of our idols and collaborators.
Yeah, I mean, it's possible that this initial discovery really is a byproduct of over 50 or 100 years of bioelectricity research. Because if you look at some of the really, really early research where people are talking about voltage gated channels and interactions, and that cancer is very electric, that's not a new phenomenon. That's been something that people have been talking about and studying and researching for a very long time. I think the discovery that Michelle Monge's group in Stanford made, where they were able to show that there was this basically similar behaviors of in cancer cells that were very, very similar to neurons, and then they actually started looking at if you patch neurons and you start to you essentially start to depolarize these neurons, what happens to the cancer cell? And they started to see that not only are they mediated by neural interactions explicitly through synaptic interactions, but they're also mediated by paracrine signaling. So when they release specific factors from neurons into the environment, or even cancer cells releasing those types of interactions into the environment, there is this network effect that's happening. And that network effect is actually really bad for cancer. So there's this reciprocal engagement that they kind of discovered that has now exploded a bunch of researchers to be like, whoa, there's like so much going on here. What are those individual sort of mechanisms? Which ones are happening through neurotransmitters? How many of them are actually synaptically integrating? And it kind of, again, creates this opportunity for a whole new host of targets, whether that be new drugs to discover or new targets, like new ways to therapeutically intervene, but also looking at repurposing. So people have been looking at repurposing epilepsy drugs now as neural inhibitors. People have been looking at retrospective studies in gliomas, for example, where they're like, well, what happens if you were on an anti-epileptic and you also had glioma, and how is their survival different? And you do see these types of differences retrospectively when you look at studies. So now people are starting to do those kinds of studies going like forward, which is awesome.
Does that mean to say that like you can imagine at some point it will become like standard of care for all people who have glioblastomas to be an anti-epileptics, or it's not that open and shut?
I mean, they're running these trials at the moment. I think if there is a significant benefit that they're seeing in them, I can't see why they wouldn't add that as a standard of care. We're talking about patients who have such poor prognosis, such poor survival. The standard of care has not changed for 25 years. So if there is anything that's beating or contributing to current standard of care, I can't see why. If the side effects are not completely debilitating, the quality of life is still very important. But if they can do that, then I don't see why that wouldn't be a natural progression as well.
That's cool. Yeah. I feel like I don't know. I usually don't hear about like free lunches like that, because like when I think of anti-epileptics, they aren't super side effect heavy. So, yeah.
I mean, another famous example from another one of our collaborators, Erica Sloan at Monash, where they're looking at beta blockers, another relatively innocuous drug, generic, widely available. And again, looking at retrospective studies on the outcomes of patients that happened to be on beta blockers, and showed pretty profound impacts, reducing the chance of metastasis from breast cancers, I believe, in a way that you wouldn't necessarily expect from something that's innocuous and has not typically been used in cancer therapy. And it's exciting. It's exciting as it's uncovering biology that we haven't thought about before. And so, yes, there's drug repurposing opportunities, but also forward-looking. What does that mean for our understanding of biology and cancer and the system itself, and how we can design new therapies as well?
On the subject of like, before we move on to like using these signals as like a biomarker of cancer itself, touching one more point on the therapeutic aspect of SOMA. Let's say like SOMA works, you're able to prevent the like cancer from interacting with the rest of the nervous system, and somehow that improves prognosis, which, given the evidence you've given so far, it feels like not too large of a logical leap. What do you suspect is like molecularly going on that is actually helping the patient? Like what about like breaking the crosstalk is actually beneficial?
So I think there's a whole host of things going on with like, there's not a single molecular interaction that happens between neurons and cancer cells. And there are interactions that happen both with, they call them pacemaker cells, which are like specific cancer cells in the network that have maybe like a synaptic integration with neurons, and then they have this gap junction mediated network that happens between cancer cells. So when we're looking at, if you were looking at neuromodulation as a regime, especially for electrical stimulation, we look at something called blocking regime, which essentially either tries to it's like a depolarization block. So you either try and stop the neuron to be able to propagate action potentials, which in turn could mean things like blocking neurotransmitter release. Glutamate is a primary example of primary excitatory neurotransmitter that has a really, really profound impact on cancer cells. There's a lot of glutamate in the brain. So being able to reduce some of those. When people have used pharmacological blocking of AMPA receptors, they see I think it's around 50% reduction in tumor volume in mice. So we imagine that there is a whole host of things that you're kind of interacting with when you block that.
Maybe a dumb question, but like why does reducing the levels of the neurotransmitters lead to a reduction in tumor volume?
Oh, they're growth factors. So,
okay.
So like glutamate and neuroligin 3 is another paracrine signaling that has picked up by gliomas that are growth factors.
Gotcha.
Yeah.
Okay.
So you're trying to reduce the growth factors essentially.
Yeah. And with regards to the actual like biomarker aspect of SOMA, you implant the device, like maybe it's post resection or before a resection. What is the actual readout that you're getting?
Yeah, so fundamentally, we're recording electrophysiological signals as well. So again, for all intents and purposes, you can think of this like a brain-computer interface. We're recording electrically, we're stimulating electrically. So we're reading out the same endogenous electrical activity of the brain as a device that's trying to read out like motor intent, for example, right? So we're looking at spike rates across the brain and more broadly local field potentials, so frequency shifts in the brain. So we're not measuring, as I said at the beginning of the podcast, cancer cells do have this intrinsic electrical property as well, and you can measure that, and we have measured that. But
sorry, actually, but before you move on, like you mentioned at some point, like we know cancer is very electric, and you just mentioned again, what does it mean that like something is very electric? Like why is cancer very electric?
They have a very high expression, or they retain a very high expression of voltage-gated ion channels.
Okay.
So when they say they're very electric, they are they hold a particular membrane potential. They depolarize under certain events. Those depolarization or hyperpolarizing events are usually occur during the stage of the cell cycle. That's what we mean by that they're very electric.
Gotcha. Okay. Sorry. Go ahead.
No, absolutely. I mean, it's important to clarify. So we are looking at this electrical activity of the brain. We use that as a proxy for what the hell is going on in the tumor or near the tumor. And because of these interactions between the tumor and its sort of microenvironment, we can use that electrical proxy to inform ourselves what's going on in the tumor. So we're not measuring specific biomarkers, for example. We're not just measuring specific proteins in that microenvironment. We're measuring the electrical activity of the microenvironment and what that means. So trying to correlate that to tumor volume or drug responsivity or just seizure activity, or maybe the aggressiveness and growth rate of the tumor, and trying to do all of that from electrical proxies.
Is that something that is going to be like pretty like instinctively? That sounds very custom from patient to patient, tumor to tumor. Is there actually some like universal properties that you can derive from like the like EEG-esque readouts that you're getting from these, or like spiking potentially you're getting from these?
We're going to find out in human. Yes, in rodent, but of course rodents are very homogeneous between mouse to mouse. Like we're going to find out in human. I will say, using electrical biomarkers as a proxy for something else is not new, right? People are doing this in other areas of medicine. People are now looking at this in Parkinson's, in pain, in depression, for example. So, in those spaces, there's still a lot of variability between patients. We expect to be able to demonstrate the same thing in humans between patients by training one model and applying it to multiple patients. That's the end goal. Yeah. Of course, proof will be when we run our long-term human studies, because these have never been done before. It's one of the most exciting things. People have done this intraoperatively. You can take a recording from a patient from a tumor while they're under in surgery.
We've been talking about like when you actually implant the device, it could be like post removal or after removal, or pre-removal or post-removal. The pre-removal setting makes some sense to me. You attach the like pins, you said, or
leads,
lead threads, right?
Not spikes.
Yes, yeah.
We're very anti-spikes, because that's old BCI.
Yes, the threads into or around the tumor itself. In the scenario where the tumor has been removed, are you just placing it simply into the cavity where the tumor was found, and you're just like trying to see if there is like any tumor cells left over? Like what's the use case there?
Yeah, in the margin. So in the brain cancer case, most recurrences, I mean, glioblastoma, again, to paint a picture, is a horrendous disease, always terminal, unfortunately, and very, very poor mortality rates. And so those patients, even you have a resection, and somewhere between 70-80% of patients are having a resection, enough to have a resection. Of those, all of them will get a recurrence again. And of those patients that get a recurrence, most of them, I think 90%, are in the margin of the where the resection was made. So that's where your sort of residual cells are. That's where your tumors start again. So that's where we're targeting first. Now, that's not to say we can't go more broad area, and we have internal programs where we're looking at this sort of broad area coverage, maybe from the surface of the brain, maybe deeper in the brain. And the idea is to get as much coverage of the brain as possible, so you can control distantly as well.
Do you? I guess like this is somewhat clear in the case where you're purely recording. You get this like spike readout. for the case of like intervention, what does the perturbation you're applying actually look like? You mentioned something about like blocking. Could you talk a little bit more about that?
Yeah. So there's a multitude of like stimulation regimes, but one of the ones that we're like really particularly interested in is this blocking regime. When we started looking at biomarkers, so when we started recording longitudinally, what are the biomarkers that we're seeing change, electrical biomarkers that we're seeing change in the brain, we saw a really interesting sort of peak in the hyperexcitability. So if we look in high gamma range, you see this week-to-week increase in high gamma activity in tumor-bearing mice.
Maybe you can explain what high gamma is.
Frequencies above 70 hertz. That's what we tend to see very distinct changes in brain activity during progression of disease. And I guess what led us to thinking about blocking was like, well, if we know the general spike rate of these kinds of neurons in a particular region of the brain, it kind of varies throughout. Computationally, we started deriving some neuromodulation parameters that modeled the neuron and essentially looked at can we stop this activity from happening? So if you generate an action potential, how do you stop it from generating it again? And what consequence might that have on cancer growth? And so that's essentially the types of perturbations that we're looking at. If you're not familiar with blocking, you can think about it similarly to how you would evoke a neuron. So if you stimulate a neuron, you evoke an action potential. When you block, you essentially stimulate at a faster rate, such that when it tries to regenerate or repolarize, it can't. And so you kind of leave it in this constantly depolarized state. It can't reach a threshold. It can't activate. Yeah. That's the kind of perturbations that we're interested in the brain and outside of the brain.
Is it fair to say that this is closed loop and that is not actively like learning about the state of the cancer electrical state?
Currently, yes. Okay. So this is sorry. Currently, no. Oh, okay. Currently, this is an open loop system. So rather than thinking of this as like a closed loop therapy, we prefer to think of this like a theranostic platform. So we have some electrical stimulation where we're treating this disease, and we have a diagnostic function where we're reading out and we present the clinician and the patient with what the hell is going on. And you can imagine that would be overlaid on an MRI to say what's going on with the tumor, in real time, really very, very fast. That's great. It's a new diagnostic weapon. Mm. Eventually, of course, you train these models and these systems to be better than clinicians so that you can make these systems closed loop. Then that's the holy grail across many aspects of neuromedicine, where you no longer have to have a second diagnostic readout and say, okay, well, how do we tune the stimulation? Where do we position the next device, the next electrode? Instead, the system's doing that for you, optimizing exactly where it's treating, where it's stimulating.
But what were you going to say?
I was just going to say that it might be helpful to understand that when we started trialing these types of blocking stimulus in the brain, which high frequency blocking is not new in neuro. Like people have been doing this for a while throughout the body, but there is a way that you can understand whether or not you're having an effect in the brain. So what we typically do is we record a segment of electrical activity, process that data, look at the frequency band, we apply stimulus, we then record again, and we see that there is a transient response that you have when you deliver this type of neuromodulation, where you can see a decrease in the high gamma range that we're interested in. And that's also how we threshold. That's also how we would work out, like, how much do we want to, do we need to increase the stimulus? Do we need to change the stimulus? Do we need to change which pairs of electrodes we might be using to achieve a certain area of activation or blocked regions of the brain? So we can use those kinds of methods of pre and post recordings to tell us whether or not we're having the effect that we desire.
Incredibly naive question on my end, but if you, it sounds like you just want to be like constantly stimulating all points of the tumor as much as possible to prevent it from ever being able to fire off an action potential.
Pretty much.
In some scenarios, yes, but we are also running studies where we're looking at dosing because there are other mechanisms at play as well, and some of those mechanisms are maybe you don't need to be stimulating and like spamming them constantly, and you can maybe perhaps dose once a day and you elicit some local biological response as well. That
I guess, like, I'm curious, like, if you're actively going to be working with like a clinician to like tune the actual inner workings of SOMA. what is the clinician like knobs of control that they're actually allowed to tune if it seems like just overloading the tumor with stimulus is like actually what you're doing in practice?
I would draw a parallel between radiotherapy. Whole brain radiotherapy is really fucking grim.
Okay.
There you go.
If you've ever seen a patient go through whole brain radiotherapy, it is gnarly, it is awful, it affects their whole brain, as the name suggests, and those patients are never quite the same afterwards. Now, clinicians don't want to do that. They do it as a last resort, and they try to use very, very focused technologies where they can hit the tumor very, very hard and spare the rest of the brain. Now, that would be the approach that we would take as well, and it's the approach that Optune takes as well, right? They focus that field, they focus the stimulation to as much of a concentrated point as they can, so they can hit that area as hard as you can, and that's what we would want to do as well, rather than targeting the whole brain.
Why, instinctively, why is there any off-target effects if all of the threads are around the tumor?
Because current spreads.
Oh, okay.
Current spreads, and
Brain is super, well, like the network in the brain is crazy. You have long-range projection neurons. You have, like, you might be affecting, the body of a neuron in one area that has a projection that's going, very, very far away. So there is definitely a network effect. One of the things that we've been looking at is how you can computationally essentially model what the affected area of tissue might be, and affected being like affected area that would be blocked. So we've actually integrated in these models of these neuron models into our computational models that essentially tell us, okay, well, this is the threshold that we need to hit in order to block a neuron X distance away. And you kind of build these like really nice balloons type shapes around the electrodes that tell you how far you're actually going to hit if you want to block neurons. And then, of course, there's probably going to be some of those as an extension of those neurons that are connected or networked somewhere else in the brain. At the same time, you're also imagining that when these types of, in glioblastoma, when these types of patients are having resections, they're having big chunks of tissue just like taken out, right? And so it's like trying to preserve function and like being aware of which areas of elegant cortex you want to try to avoid, so that you're not inhibiting movement or inhibiting speech or inhibiting critical functions, and trying to like design the stimulation parameters or the way that you might activate those electrodes. Where should they be in order to avoid those spots? And you can kind of do that by taking the MRI in as well.
That makes sense.
Yeah.
Actually, I'm curious. Like you do mention that like SOMA is MRI like safe. Mm-hmm. Why is that important or particularly useful?
It is absolutely critical for oncology. MRI is not going anywhere. It's the gold standard imaging technique, and it's used in the brain more than anywhere else. In glioma cases, they ideally want the patient to be having an MRI every 3 months. So if you induce a device, the body that is non-compliant with MRI, that's a massive problem. That's one thing. The second step is not just inducing compliance into the device, but making sure that device doesn't cast any artifacts. So MRI relies on magnetic fields. And if you have a magnet in your device, large chunks of metal, then that will affect the MRI image. You start casting shadows. Your clinician's not going to like that. So we've spent a lot of time engineering this device and using technology in this device to overcome this issue.
Is like external from the actual like inner workings of the device with regard to therapeutic interventions and like the biomarker readouts. I am curious about like the practical use of this device and like a clinician's workflow. What will it look like? Like you're diagnosed with glioblastoma, you immediately have this put in, or what?
We're gonna work our way up through patients, right? So our first patients will be the most sick recurrent patients who are probably coming in for their second surgery at this point, and the device will be left behind. Then we'd be moving towards newly diagnosed patients, patients who maybe I can walk through what that generally looks like for a patient. Patient would usually present with perhaps a seizure. Fit, healthy, 42-year-old man or woman has a seizure. They go to A&E. The A&E doctor will say, I think you should have an MRI. You have an MRI, they spot the tumor. Pretty quickly, you're brought into surgery. Almost, within a few weeks, ideally, you're brought into surgery for resection. Our best clinical access point would be right there. Like leave that device behind at that first surgery. Patient will then have radiotherapy and chemotherapy, temozolomide usually. Eventually, of course, we want to work our way up through that, and we want to be the top of that pile, right? Yeah. So with radiotherapy, with chemo, we want to be right alongside it as well.
When it comes to let's say you run a clinical trial with SOMA, the thing that I would be instinctively curious about is that the patients who are most willing to have this device put in are also the sickest, and potentially the device might not be able to do anything at all. Is that at all a concern?
I mean, no.
Okay.
we will have done a lot of work preclinically, right, to validate this technology, and we're adopting some of the lessons from Novocure as well, of course, that is now very clinically validated. It's been out on the market for 20 years, and we're adopting some of the lessons from that. Now, of course, early feasibility patients are always signing up for a clinical trial, and we won't be signing those patients saying we are going to guarantee you that this is going to end your disease and you're going to live forever. That's of course not something we can do, just like any other drug or device trial. So of course we're going to be working within the bounds of ethics of clinical trials as well on that. But there's a hell of a lot of work that leads up to that, so that we're confident that we're going to be having a clinical effect, therapeutic effect.
The one thing I did want to ask, and that's something we've talked about in the past, is whenever someone outside the bio field hears about bio, their first thought is oh, Michael Levin. If I put my Michael Levin hat on and look at Coherence Neuro with that hat on, my thought is well, if you put SOMA into a glioblastoma, why can't you just reprogram it back into a normal neuron? Because all cancer is membrane depolarization gone awry. What? Obviously, that probably isn't true. But what is your view of the Levin-esque understanding of bioelectricity?
I'm going to hold back for a second.
I think the way I interpret Levin's interpretation of what's going on in cancer is essentially that cancer is maybe mostly influenced by external cues, less so a genetic sort of abnormality. And I think that aligns a lot with the way that we've been building this technology, and how we would use this technology is we're trying to influence the environment, given that that's a dominant factor for how these diseases are able to thrive. And if you imagine that, if you can believe that cancer cells are able to modify themselves to thrive in a particular environment, it shouldn't be so far-fetched or impossible to believe that the same can be said in reverse. I think that the challenge against some of this thinking is that cancers are normally diagnosed at a really late stage, and at that point you have a significant number of drivers or mutations that have happened, and it's saying that you can essentially nudge these cells back into a healthy state is a bit of an oversimplification. However, I do think that with if by using something like a bidirectional interface, where we're no longer just relying on what do we see in a cell at that specific point in time, what is the snapshot information, what do we see at day 1 or what do we see at day 5, and we miss the entire thing that happens in between. I think that there is a lot of information that we can get out of that longitudinal data. So what happens to that cancer cell or that environment over the course of its evolution? And we don't have that yet, and that's exactly what we're trying to build. And I think it's not also infeasible to think that these types of devices will be able to have that single cell resolution at some point. So while I don't fully buy just yet that you can just nudge these cells back into a healthy state, I don't think that it's so far-fetched. I think that if we understood what was happening that makes them kind of change through time, which we still don't know, perhaps if we listen to them and we read from them and learn what's happening, I don't think it's that crazy that we can start thinking about, well, what kind of nudges do we need to make to put them back into a healthy state. So not super crazy.
Yeah. So the argument is at the very start, there is genuinely its membrane potential gone wrong, and then driver mutations are acquired, and then it's irreversible.
Well, I think that in a simplistic view, you could say that there is so much going on in a cell, right? It's like it's an oversimplification to say that one single voltage channel is like driving this entire process. I think there are multiple things going on, multiple different potential, membrane potential mediated interactions that are happening that drive the change in DNA or, whatever else it might be that say, okay, now change this expression, express more of this protein and whatever else so that you can leverage the environment. Like, I think that, like, as they're growing, that only happens exponentially. So you're having so many more of these mutations happening. And as the environment changes, they change again. They're really, really clever at like figuring this out. So I think that because we catch it after so many things have happened, it's very hard to work out, well, how are you going to go back and change 15 or 16 different steps that have changed with one single application?
In a highly heterogeneous tumor environment that now has 40 different cell types or however many.
Yeah, and I don't think you can also make the claim that if you stimulate that environment, or let's say you try and target just the cell, that there's no consequence that happens to the neighboring, non-cancerous, healthy participating cells. You're going to modify them too. So like, how do you design a protocol, or how do you design a system that essentially only targets those specific channels, for example? Mm-hmm. If you look back at about, I think it's around 2010, there was a really incredible article a review article that looked at what happens in cancer cells, what happens to the membrane potential, or what happens when they depolarize just before they enter a certain stage of the cell cycle. So it's called cell cycle mediated membrane fluctuations. And it's like there's so many things that are involved in that process. And so I don't know how you could. It would be difficult, I think, to really specifically target like one specific version of the ion channel that can mediate that change. So I think it's complex.
I'm going to go one level higher. Ion channels are important. Membrane potential is important, and our best chance to mess with it and target it are by using, electrical biological interfaces like high-density BCIs. Yeah. That's exciting. I think there's a lot of potential there. I don't think we know yet what the downstream effects can be, because a lot of this has been done in a dish. A lot of this has been done maybe in simplistic rodent models, and not a lot of this has been done at the network level and a single cell level in a human brain. So I think it's exciting. I think there's a lot of potential. Jury's out on whether you can fully reverse cancer back to a healthy state.
At least at the for like SOMA-like devices that have been tried in, I'm assuming like mice models. Mm-hmm. Has there been anything more complicated than mice, or is it just mice?
In
neuromodulation for cancer.
Mice for cancer. All of our safety work is done in larger mammals.
Okay.
The cancer models, I mean, as you know, is coming from the cancer world. The cancer models in larger mammals are less useful, let's say. Spontaneous models in some companion animals that can be used, but
how well does this work in mice?
Sorry?
Like neuromodulation for cancer.
So in pharmacological settings, like what people have shown is around 50% reduction in DIPG, so pediatric glioma. So that's like essentially our target. We've been looking at how like different types of stimulation parameters in glioblastoma versions of like models of that. So yeah, we're still working on that right now.
Is that for like mono treatment, or is that combined with something else?
We do both. Okay. So we're looking at both combi treatment with the standard of care, so which is temozolomide, and we also do like standalone treatment with a host of different neuromodulation parameters.
Actually, this is, I guess, something we completely did not discuss at all. As to like potentially SOMA is useful for not just like even if a patient is going to live only 2 more months, is SOMA useful for just like reducing symptoms? Is there a good argument that could be made there, or is it like iffier?
I think that there has been there's work that has shown that neuromodulation can be used to reduce seizure activity as well, and electrical stimulation can be used to reduce seizure activity. There's a hell of a lot of work that's been shown that you can reduce pain, and you're targeting some of the same nerve bundles that sensory neurons are traveling down. So I think it's very likely that we will end up reducing seizure burden, pain burden, etc. from this. But again, we can't speak to our rodents, so we'll have to find out when we do our interop and safety work in humans.
My impression is like, are you able to just like automatically take advantage of all the neuromodulation literature that's out there when using SOMA, or do you need to build up your own corpus of knowledge because it's a brand new device, it's being used for cancer, it's at the site of where cancer just was?
I mean, both, right? So we massively leverage elements of Optune and Novocure and elements of the neuromodulation world. Okay. And so this isn't coming out of thin air. There is a body of work that's been around for in the neuromodulation world for 60 or 70 years, and we leverage a lot of that, looking at how these electrical stimulation routines do affect biology and neural firing, etc. So we are adopting some of that and applying it to new diseases. Yeah.
The only thing I would add to that is that especially on the device, like product development for the human device for SOMA, there is so much neuromodulation, electrical neuromodulation devices that exist that we can 100% learn from. To the extent of how do you characterize the electrodes? This is really, really important to make sure that they're safe. There's all of that literature is directly relevant and useful that we use all the time.
Do you, well, actually, this is more of a broader question, but if it turns out that cancer broadly interacts with the nervous system, why go after glioblastoma specifically and not just, I guess pan-cancer would be too ambitious of a goal to start off with, but alternatively, maybe why not go at a cancer that's perhaps less fatal and a bit easier to work with?
I think pan-cancer is the right amount of ambition.
Okay.
We just as a slight side quest, we do want to go pan-cancer with this, and I think what we've seen historically in cancer treatments, the ones that really move the needle are the pan-cancer approaches that tackle some fundamental mechanism. So it's cut the thing out, right? Yeah. Is one of the most effective approaches for cancer treatment. Burn it with radiation is one of the most effective treatments. Chemos, immunotherapies, things that do affect lots of tumors. And we're going for the same thing with this. We want to build devices, one for the brain, one for the torso. And we want to go after as many solid tumors as we possibly can. Any of them that we see an effect in, we'll be pushing forward. Why start with the brain then is the next question, because it's hard. There are a number of reasons for this, both economic and technical and cultural. So, number one, Novocure has set the setting for the use of electrical devices in brain cancer. The FDA, regulators, and payers are comfortable with the use of electrical stimulation devices now in glioblastoma, and that's a big cultural moment for these kinds of devices. Clinicians are comfortable with the use of these devices and use with physical modalities of treating the disease as well. And number two is the surgical elements. For our first devices, now this may not be the case forever, but for our first devices, we're implanting, we're going inside. So we want to be looking at diseases where surgical intervention is not uncommon.
Oh, okay.
So again,
bring the barrier right down.
70-80% of patients are having surgery. The risk floor is established. Yeah. Leaving something behind is marginal risk there versus, trying to justify, with many of these neurotechnology companies, trying to justify new surgery for a patient, the risk reward starts to get a bit complicated. Yeah. For us, we don't have that issue. And then the final large reason, there are many, but the final large reason then is how many therapy options out there, and what are the macros looking like of new interventions coming to those diseases. Now, glioblastoma, unfortunately, is not a pretty picture when it comes to what's on the horizon. The standard of care has not changed for 25 years. Optune is probably the most transformational thing that's happened in those 25 years. Outside of that, there ain't a lot of hope. There aren't many clinicians singing the praises of other technologies coming online over the next 10 years. And we want to be at the top of that pile. We want it to be, again, resection, radiotherapy, chemo, and us. And that's not quite easy as an equation for, say, breast cancer, for example.
Is it, you mentioned that Optune has kind of paved the way for medical devices to be used. I'm assuming by virtue of this being invasive, there will be some new territory that you have to poke at. What is that new territory? What are the logistical and regulatory challenges ahead?
Regulatory, we're not scared of. Regulatory, we will get this device approved. It's very likely that we'll get breakthrough designation for this device. I'm not concerned about that. Reimbursement is your biggest threat and challenger in devices always. We need to be designing trials, we need to be designing the device, we need to be designing how the patient interfaces with these devices and how the clinician interfaces with these devices in a way that also makes payers happy. And again, Novocure has laid some of that groundwork, but there will be, of course, different costs. There will be different costs involved in like the surgery with the patient, how the surgeons are interacting with the device, how the external components are being supplied to the patient will differ, and we need to design trials and a go-to-market strategy that lends itself to that well.
This is maybe related to the actual like implantation process itself, but do you need like a Coherence employee alongside the surgeon, like helping guide how exactly the device is put in?
No, I mean, it's a good question, but no. Okay. We've been designing these technologies alongside clinicians, neurosurgeons, neurologists, to make sure that we're compatible with what they already do. So how they plan surgeries, how they implant devices, so that we're not having to build $100 million robot.
Yeah, the Neuralink way. Yeah.
To do that. And there's obviously some incredible engineering that's gone into that. But right now, we want to get into patients as quickly as possible. They don't have much time, and we want to get there fast. And the best way to do that is by giving a device to a clinician that requires minimal surgical training to start implanting patients.
Yeah, it actually leads well into a question that I've had for like 30 minutes now. As to like, what are the axes of improvement that are on the table for SOMA to be improved upon?
Size is critical. Smaller you can go, the wider your patient population, and the safer these technologies are. So everyone's trying to move to more and more minimally invasive approaches, where eventually, as Elise said, you're going through some very small, single-digit millimeter access point into the body.
Is it like if it like the size it's at right now, or maybe like at a certain level of size, you're limited to like the most severe, like largest tumors?
You would be. I mean, we're already very, very small. So our device is about half the width of a Neuralink device, which is compatible with its standard burr perforations into the skull, and they'll do that for like a biopsy, for example. So
is there a good visual indication of like a nail?
Thumbnail is about
thumbnail. Okay.
Yeah.
Okay.
Yeah, thumbnail.
Yeah.
Okay. It's about the hole.
Okay.
Now, there are other improvements, of course, in the device, power efficiency, for example, electrode coverage, all these kind of elements where, eventually maybe you want high density electrode coverage to get more and more precision in your stimulus and recording. There's, of course, always improvements to be made.
Yeah. Big one for me is access. So if you, right now, if you have, one of the limitations that you might see across a lot of neurotech platforms that are going out today is like how much access of the brain can they get? Let's take, like, Neuralink has a really high density multi-thread device, but they're all going into a specific region of the brain. One thing that we've been really focused on is how do you get multiple access, like how do you create a device or a platform that can get an access to the front of like somewhere here versus somewhere in the side of the head versus somewhere at the back of the head, so that you can access multiple areas, but your surgery is still very, very minimally invasive. So by going and trying to make everything and shrink everything down really small, you can imagine not just having one of them. Maybe you can have multiple of them. And now not only are you accessing this specific region of the brain, but you're accessing this region and this region and this region, or across hemispheres to see maybe it's migrating across. And so that's like something that is also pretty hard, but quite interesting on our end.
I'm actually, what's the if a tumor is at one side, why would you care about like tumor is at the occipital cortex? Why would you care about what's going on in the frontal cortex? Because
they're actually like what we were talking about earlier when we were talking about these network effects that happen in the brain, is you have a crossover point in the corpus callosum where you have neurons that are, or like motor activity that might be happening on one side is actually
but they're like projecting on over. Okay.
Yeah, they're projecting across. So you can imagine that, like, let's say you have a very diffuse tumor that is making its way across the brain and actually going to like inject itself into the other hemisphere. If you long down the line, if you had a device that had in the primary side of resection that you put some electrodes or some probes into that region, but you kind of know that they are going to at some point migrate across, you can also put an electrode there and you pick up the signals before that they start moving across and maybe start stimulating earlier on that side of the brain.
Wait, sorry, what do you mean by migrate across?
So when it's not uncommon for very diffuse tumors to move from one hemisphere across to the other hemisphere.
I did not know that.
Yeah.
It's terrifying.
It's really terrifying. And you can't see this in MRI for diffuse tumors because they don't pick up the contrast. You can't see them, which is a problem. But you can record them, and we know that we can record them. So if you knew that was happening, again, this is like in a future sort of scenario, but if you were able to implant in multiple regions of the brain across hemispheres, you can start to actually record when that is happening, and you can pick them up from long range projection neurons as well. So we could start recording that information and also start intervening at a much earlier time point.
Is it obvious how many like SOMA-like devices you would want in a glioblastoma patient's brain? Is there like a max, like 7 of them is like enough to cover all the important spots, or?
It would be entirely based on their MRI. Okay. So you would essentially, in the pre-op setting, you would take their MRI. The surgeon will know the extent of resection that they will likely be able to perform, and you would pre-plan, and you would have a software essentially that tells you position the electrodes in this position to get this coverage. That would be how we would do that.
Okay. This is returning back to an earlier thread about like all the mouse discussions we've been having. How big of a concern is translatability from a mouse platform to pig to human? Is like membrane polarization like a pretty well conserved phenomenon across all life, or is it like kind of case by case basis?
Particularly in neurons, it's very well conserved from mice to pigs to humans. We started almost all the way in computation, so in silico. Then we went into in vivo models. In vivo models for cancer, or mice models, I should say, are the.
Wait, sorry. What does in silico models look like for neuromodulation?
So you model the neuron using a Hodgkin-Huxley model. You can computationally, mathematically build that model. You can get that model to then generate specific spike rate. Those are like quite well characterized depending on the region that you are in the brain, and then you can start applying stimulus in silico, so in computationally. That kind of helps you with selection of what kind of stimulation parameters you think might work best for the region of the brain that you're in. Then you go into mice models. These mice models, as you will be aware, are the most like relevant models that we can use for oncology. We use orthotopic models, xenografted models. We're taking human cells, we put them in the brain. It's quite a hard model to do, and then we take a device that is already very difficult to make small in humans, and we make it 10 times smaller, and we put it in mice brain. And we do a number of tests over a short distance, like short durations, to work out what the optimal stimulation parameters, what the effect of those will be. Then we go to large animals. We've done large animal studies. We've been able to show the same suppression effect in large animals in healthy brain. And then the natural progression again from that is to go into humans. So we just got approval to do our first in-human study to try these stimulation parameters in humans. So far, the trajectory seems as best as we can possibly expect.
That's exciting. Does that translate to a phase 1 trial, or is that something?
It's our first in human safety work. So it's not a chronic phase 1, but we'll be doing recording, mapping, and stimulation safety across the brains of patients.
Okay, and this is for glioblastoma patients.
For glioblastoma. Yeah. And that will lead then into our
exciting.
Yeah.
Why does this not exist today? Why doesn't every glioblastoma patient have this?
There has been a lot of innovation in neural implants over the last 20, 25 years. Miniaturization of electronics, better powering methods, new electrode materials and lead materials. There has been a hell of a lot of innovation, things that didn't exist in the early 2000s, frankly. On top of that, there's obviously with Neuralink coming to the table in BCI, there's obviously been a lot more focus on the use of these kinds of technologies across diseases as well, many diseases, and a lot more cultural acceptance from clinical centers as well to adopt them. So I think that's one of the reasons.
Yeah, I also think that for us, like the scientific underpinnings of these interactions are still very new. The discovery of bioelectricity is not new, as we said before, but these neural interactions that are happening and observed in cancers are really new. So I think that in combination with the ability to miniaturize technology and get them implanted chronically, and record and stimulate these environments for patients who have literally nothing else. It's like, yeah, those have been the limitations before now.
And to underline how new that is, we present sometimes to academic cancer groups or cancer neuroscience groups. We show our mouse setups that we've developed as well. And it's kind of a little bit mind-blowing for them that you can do high-density neural recordings across the brain of a mouse over months, 6 months, 4 or 6 months. These are technologies that, again, they haven't quite existed in that way. They didn't exist in that way 30 years ago. And so, we are really at the early stages of that.
When you go to what is the AACR or something and present your results, it seems like this is such an interdisciplinary field that there probably can't exist that many people in the world who really understand the intersection of cancer and neuromodulation, and whatever other field you guys are intersecting with. Do most people seem convinced today that there is something here, or there's still some skeptics?
I think if there are not skeptics, you're not working on the bleeding edge. I think you don't want people to agree with everything you're saying. Mm-hmm. Our interactions with clinicians and cancer biologists, I would say it usually goes like this. I'm not sure. And then we show them data, we talk through them, we show them devices, we show them work. And then there's a big buy-in. Then people are very, very excited. And I think they see the same things that we see. By the way, I had the same interaction. I come from a neurotech background, neuroengineering background. I was working in spinal cord and spinal cord injury for years. I had the same response when Elise showed me this sort of 4 years ago. And it took me a while to digest the papers and read the research and then go oh man, why is no one looking at this? Why is no one there's so much opportunity here. I would do this device and this device and this device. And now we're sort of having the same effects from clinicians who then start saying, well, hang on a second, I would this is how I would design the device to do this, because there suddenly becomes quite a lot of buy-in. So I think we're just at that sort of takeoff point right now, and I think we're going to see a lot more attention clinically and probably some companies as well take off, and we're excited about that as well. Similarly,
when Elise showed you these results for the 3 years ago?
4 years ago.
4 years ago.
Yeah.
Do you think that was the only moment a company like Coherence could have been founded? Or was it just you were kind of right place, right time to actually discover this information and put all the pieces together and think there is unmet need here that's filled very cleanly by this device?
I felt like I was very lucky because I was really interested in, actually, I was kind of brought into the PhD to look at a drug delivery implant. I knew nothing about glioblastoma. I knew nothing about cancer in general. I'm an electrical engineer. And so I really, really wanted to understand the problem. And when I started looking at this problem, it's horrific. And I started looking at, what is the potential of a drug delivery platform, an implantable drug delivery device going to offer here? These cancers don't respond to these drugs. And maybe you can repurpose drugs that can't cross the blood-brain barrier, and that's one advantage. But they still just manage to evade these drugs and kill patients. And then I heard about Novocure's work and I was like, absolute bullshit. No way. This doesn't work. This doesn't make sense. I built a platform to try and replicate the work because there's a whole history to that. But I was like, I'm going to figure out what's going on here. I'm very curious. And I could not disprove it. I tried. And I kept seeing what they were showing in their data. I kept seeing that these cells would halt when you would deliver this type of electrical stimulus. And my PI, George Meliaris at the time, we were talking about, well, what happens? There's a whole, there's an infinite parameter sweep that you can do here that looks at uncovering how do cancer cells behave when you put them under certain electrical stimulation parameters. And at the time was when the work of Michel Monge's lab came out. It was 2019, I think it was. And one of my other advisors had pointed me to this work. So I was quite into the membrane potential. I was like, maybe that's what TTFs are doing. They're modulating calcium ions or whatever else, or calcium modulators in the cell.
Levin's right.
I was like,
Novocure just don't know it.
And I was convinced that was what was going on with Novocure. And
so at that time, the mitotic spindle theory was not proven out?
It's definitely been hypothesized.
Oh, so like even today, it's not like known for sure.
It's there's been a lot of evidence that suggests that that's what's going on. Yes, but I think from an engineering perspective, it was not making a lot of sense to me. You've got like a very weak force acting on a very strong force happening inside this protected barrier in a cell. It was like I was struggling to fully comprehend it, but it worked, and under certain directionality. Actually, it was a piece of work that we worked on together. It does work, and it has
watching George, still waiting for that paper to come out.
And under certain, if you can control the direction of an electric field, you have a really profound effect on TTF. That was what we found in some of the work. But Michelle's work came out, and that was when I was like, that was my holy shit moment. I was like, I was working in a lab full of like these amazing like people doing neurotechnology, making wearables, making implants, making spinal cord stimulators, everything that you can think of that interacts with the body. Like our lab was building it, and I was like this weird person doing cancer in the group. And I was like, this paper came out from Michelle's group, and I invited her to give a talk to our group, like totally fangirling. I love her work, and I was just like, there is such an opportunity here. I was like, initially it was actually more on the recording side. I was like, these neurons are interacting with these cells. You can read this. Like we've always struggled to get the single cell resolution, to reconstruct that because it's very noisy in the brain. You're getting all of the neurons are kind of telling us most of the information that's going on, and the cancer cell signals maybe are a lot weaker or at a much lower frequency. It's like if we can listen to the neurons, like that's amazing. That was kind of what I tried to. That was when I went to Ben and was like, let's use your device and let's put it in here and let's listen to what's going on. And we were like, Novocure works. Like we'll stimulate that using that. And now it's like, well, fuck. There's like way more opportunities that we can do now because look at all of these interactions that are happening, and all of them are like a function of neuromodulation or something that we can modulate with neuromodulation. So
at the time you were working on novel devices for measuring
and stimulating for spinal cord injury, brain interfaces and spinal cord interfaces.
Okay. Yeah. This super cool story. This leads well into a question I had that we chatted about previously about like why build your own device for this, and why isn't there like some standard, like there is like Utah arrays that you can like hijack and like do you just use that? You don't have to build your own thing. But it doesn't seem like anyone does that. Everyone hand rolls their own thing for their own purposes. Why is that a practice in this field?
It's a good question. There are white label device manufacturers where you can take a device and stick it into an indication, neuromodulation indication, stimulate some nerves and do your thing. But there are indications where it really does make sense to create your own device. You need a certain density of electrodes. You need be compatible with the clinical workflow. In our case, MRI. Like we can't just take any of those off the shelf devices. You can't just stick a Neuralink device in a cancer patient, because they're going to have an MRI, and that the magnet in that device is going to affect the MRI.
And there's no off-the-shelf device that is also MRI transparent?
Okay. There's not.
Definitely not transparent. It's really hard to build.
And so it makes sense for us to design sort of purpose-built devices for the treatment of these diseases that we're interested in, rather than taking off-the-shelf devices. Now, that's not an easy lift. It takes a lot of engineering effort. We have a very excited but exhausted engineering team who are doing this. But it's necessary for us.
Returning back to the original story of Coherence, like you showed Ben your work, or like you took Ben over to show your results. You decided to form Coherence 4 years ago. What were like the initial set of milestones you had set up to like prove whether this is a real thing that could be scaled up into a company?
For us, it's, do clinicians and patients want this? It's very easy for engineers and scientists to start creating things that they like, that are passion projects, without actually speaking to the end users. We see this all the time. But we went straight out and started speaking to clinicians, and the pull is huge. I don't think we've spoken to a single clinician that has said, I wouldn't use that. Every single one is like, tell me when I can run a trial with this. I want to run a trial with this sort of technology. Then we've gone out and started speaking to patients, and I've become friends with a number of glioblastoma patients. And I just hang out with them and drink coffee with them. and watch them interact with the technologies that they're using. And I would say it's a pretty universal, this disease sucks and my options suck.
Yeah.
And I'm using this piece of technology and this piece of technology, and it's horrible, and I don't like it. And if you tell me right now that there's something better, I will go back and get another surgery. That's a big barrier. People do not like going in for surgery. So getting that clinical and patient pull was huge. Now, how do we transform that then into like tangible milestones? Then it's can we build the technology that they need? We've been doing that now for almost 3 years, running safety animal studies. As Elise mentioned, we're then doing a first-in-human safety study. But the next piece is going to be, what does our early feasibility look like?
Yeah.
Get it in patients, first 10, then 100, then maybe 500, and show that there is a meaningful clinical, therapeutic, and diagnostic benefit to these patients.
I'm curious, these glioblastoma patients that you're friends with, like one device they interact with is probably Optune. I've heard kind of sucks because you have this like constantly heated device near your head 24/7, or I think like above 18 hours a day. What other technology do they have that they like potentially use to help their disease?
Not a lot. There's some things on the horizon that people have started experimenting with, that people have been on in trials with, looking at ultrasound sort of type devices, blood-brain barrier disruption type devices, continuous drug devices. What are they called?
Convection
enhanced delivery devices. They're all pretty. They're not great.
Yeah, there's like no silver. I guess you mentioned like there's no silver bullet.
There's no silver bullet, but it's also just like the quality of life and the patient is impacted. Okay. I don't want to sit here and talk crap on Novocure. Like I'm really happy that company exists, and I'm happy that technology was created for those patients who are in desperate, dire need. And the engineers, the scientists, the people that run Novocure, like kudos to them for bringing like a new novel technology to those patients who desperately need it. And of course, those patients are using it because it is extending their lives. But there's so much more you can do. Like there is so much more you can do to enhance the quality of life of those patients who don't want to spend the rest of their lives like traveling with companions to align stickers on their head and being affected by skin rashes and having the pain that's associated with all of that. Like there is much more we can do for those patients.
When back to like the creation of Coherence in the first place, you talk to the providers, you see there's demand, you talk to the patients, you see there's demand. Now it's time to raise money. And Coherence feels like in this weird place where the thesis is so strange that there is like not really many investors I can imagine off the top of my head who are like instinctively like they did their PhD in this area and they understand what you're talking about. How difficult was it to raise money for a thesis like this?
They'll come around. They'll see what we all see, and they'll realize that how large the pan-cancer opportunity is. Yeah. How hard was it? Both hard and easy.
What was your seed? Or
we did a pre-seed in the end of 2020, end of 2022, early 23, which was about $2.5 million. And then we've just very recently closed our seed round, which was another $10 million. I think the investors that we've brought into that are, again, they follow the same trend that scientists, patients, and doctors sort of all have with us. They're sort of like cautiously, skeptical at the beginning. Hang on a second, how's this work? And then go in a very big way, get very like interested, obsessed, both on the therapeutic opportunities, but also on these like data creation opportunities. So we're living in a world now where there's a lot of like AI bio companies out there, and they desperately need data and novel data sets that are showing progression of disease and sort of novel insights from human biology. Is exciting to a lot of our investors as well. A lot of people are excited by BCI, but they're all looking for what's going to be the killer application. And so I think when people get that impression of us, they go all in.
Yeah. Speaking of TAM expansion, one market is pan-cancer, but I imagine there is a very reasonable logical leap you can make that like, membrane potential is probably important for a lot of diseases. Is that true? Like, could you make a reasonable argument that like, you don't really need to do like these like 5-year-long Alzheimer's readout, like disease progression readouts, you can get a decent proxy from electricity readouts. Is that at all like an argument that people are trying to make?
It's an argument that people are trying to make. It's not something that we've done inside the company, but people are exploring electrical and other physical stimulation modalities in Alzheimer's. People are looking at recording readouts for Alzheimer's, Parkinson's, other neurodegenerative diseases. And then, of course, there's a whole host of neurological disorders that people are looking at, both electrical readouts and electrical stimulation in, and other systemic diseases like diseases of the immune system and other things as well.
The nervous system is involved in everything.
Yeah.
So I feel like it would be not a surprise to me that these types of interactions you can pick up. You can pick up a whole host of things going wrong just by looking at nerves or neurons.
Mm-hmm.
Is there any like convincing evidence today, like outside of cancer, if someone gave you a few $1 million to throw in another indication on top of what you guys are already doing, what would be the next thing?
A company that's just launched that's looking at nervous system, targeting the nervous system for treatment of asthma and COPD. Before I did my PhD, I was actually working in lung diseases, drug delivery for lung diseases, and I actually think that's a pretty big opportunity. Chronic diseases, many patients are not managed particularly well. A lot of hospitalizations. There is evidence that you can stimulate certain nerves to relax the lungs, to bronchodilate, and closed loop opportunities as well when predicting when someone is about to exacerbate. I think there's a big opportunity there.
Oof. Chronic stress or TBI?
TBI is interesting.
Why TBI? What's actually? I could fabricate an intuition for myself. I'd prefer you guys give one to me.
I think traumatic brain injury is really, really interesting and has similar attributes to what you can leverage from glioblastoma. A lot of the time when someone has a traumatic brain injury, they're already going in to put something into the brain, usually a shunt, or there's like an obvious access point, which I always think is the biggest barrier to entry right now. Until this becomes more mainstream, that's the biggest barrier. So that's like an obvious one that stands out to me, is like they're going in and doing something already.
Proven biomarkers.
Yeah, there's proven biomarkers. Yeah, spreading depolarizations are, and again, you can do similar types of strategies to suppress activity there. So, I also think what's really interesting there is I'm really interested in the data side of all of this, and how these types of issues, diseases, degeneration, whatever else evolves. And I think what would be really interesting looking at TBI is looking at how you can watch a brain go back to its normal, like, healthy state, and what type of biomarkers are we picking up that gives us that implication, or that gives us that idea that something is going right, and how do you steer that? And so that's really interesting in TBI. So that's why I would think that chronic stress, just because it's regulated by adrenergic signaling, so you can just jump on the vagus nerve or something else.
This other indication.
Yeah, that makes sense. One thing I have been curious about is I think I interviewed Hunter Davis a few 2 months ago, Until Labs cryopreservation guy. His company, I think, shares some similarity with yours in the sense of like it's wildly over-disciplinary in a way that very few other companies in the world are. And he had some interesting thoughts about like how hiring works in companies like that. I'd like to get your view, both of your philosophies on like what makes for people you want to join Coherence.
You want to kick off?
Ooh, yeah. I think probably curiosity and taking lessons from other industries. So some of our engineers come from like the robotics industry. Some come from the med device industry. Some are scientists from completely like outside of like cancer neuroscience. And then we also have cancer biologists who really know cancer, but also know immunology and also know neuro. So we kind of look for people who are like, I guess, experts in their domain, but also that have demonstrated interdisciplinary overlap with multiple things. So robotics is a really nice example of that, where you have like mechanics and you have electronics and you have like spatial interactions and those types of things. You have to all consider those into your design. So that's like a nice example. The scientists are some of the funnest ones to find, because a lot of them are coming from the neuro background. We get a lot of interest from neuro because that's the kind of talent we seem to attract. But when you introduce them to this concept of these interactions that happen in cancer, like people's minds just like massively expand, and that watching that process when you start going through that in the hiring or when you bring them on board, and how quickly they go from never hearing about it ever before to like being so on board, building these, like designing these crazy experiments to try and uncover some new neural biomarker, and like the way that they are like so bought into that, that's like been really, really cool to watch. Especially when you have this crazy idea many years ago, like that no one's ever heard of, and you've got all these people that are like super pumped about that discovery and want to build something that interfaces that discovery. That's been really cool.
Yeah.
Mostly I'm looking for interdisciplinary backgrounds.
Yeah. Code switching, as Elise says,
Sorry.
code switching and across disciplines, super important. And I guess it's the same as a lot of like very deeply technical companies. It's like your ramp of being learn, right? How steep is that? How steep is yours? Because we have electrical engineers that need to come in and learn biology really, really fast. We have computational neuroscientists that come in and need to learn how to run, what be adjacent to clinical studies really, really fast. And BCI and neurotech is a field that has so many touch points. Yeah. Electrical engineering, neurobiology, to the sort of stuff that you do. And it's hard to find people that are willing to spread themselves across that many fields.
What do you think is the rarest skill set to find and or to teach?
We know this because it's the person we're always trying to hire. It's like very, very good electrical engineers and embedded systems engineers. So hard. They're hard to find. I think it's a feature of the Bay Area. I think
there's not that many hardware people here.
Well, I think a lot of the electrical engineers that have come out of Stanford or wherever get attracted by the tech industry, and they're good. They're often good programmers, and so they go to Google or Meta or wherever. And we need them when they're often like at least a few years into their career. They've got a few projects behind them, a few product cycles if we're lucky, and most of them have gone into tech. So we're bringing them back into hardware. It's tricky. I think we're seeing a shift now. Hardware is kind of hot again, right? So maybe a year or two there'll be a bit of lag, and then we'll see more hardware people that we can bring into the fold. But it's always the positions that we're fighting most for.
I think it's like I think some of the most talented people who have ever I think joined the companies I've been a part of have been like ex-engineers at like Cerebras or like Uber or like these the big SaaS companies. What is like the big company in your field that you wish you could just pull all the engineers from to come work for you guys? Is there one? Like Neuralink?
I think Neuralink could be a good one, because given that they've just taken strides in being the first ones to take both a BCI, like a high density BCI, and a robot into trial in a really short period of time. I think there's a lot of things that those people would have learned along the way that could definitely be leveraged at a company like ours. I think there's a challenge of when you're trying to do something really new, but it's also a regulated technology. There's this balance of being able to bring in people who really know how to build medical devices that are not scared of things that are new. So that's a really hard balance to find. There's no company, maybe apart from Neuralink, where that exists.
I think the last question I have is, if you were given $100 million equity free to push this work forwards as fast as possible, but you had to spend it within the next year, what would you spend it on?
I have 2 answers for that. Can I give 2 answers? Can I have 1.5 answers? I'll give 1.5.
You can have as many answers as you want.
This technology exists. Yeah. The technology that we're building fundamentally exists. There are no science challenges, right? This is like an engineering optimization piece now. Being able to get those technologies into as many human beings, as many cancers as possible, we could build such insane data sets. We could build such incredible real-time, real-world data sets that would, I think, blow a lot of people's minds of what you can access from that data. You just can't run that many trials all at once, right? If you don't have 100 million equity free cash. If you're offering, I will take it. The other super exciting thing would be, fab floor, engineering integration floor, clinical scientists clinic in one building.
Like everything in-house.
Everything in-house, including like, you know, a clinic, like a neuro-oncology clinic would be insane. I think you could do that for just about 100 mil if you did it maybe not in America. That would be incredible. I think you'd have to put the clinic on the bottom floor, actually. You wouldn't have the surgical theater on the top floor. You'd have in the basement. That would be, that would just be insane. Being able to like highly iterate, build devices, build them in your own clean room, validate them, get them in patients like really, really fast, and start, you know, running studies, collecting data, and becoming that, you know, hub of those studies.
Why does it matter? Why do you care about having like a clinical oncology suite inside the building?
Just so you have some control over the functions, the implants of the device, the same surgeons, the, you know, like quick readouts connected to your. When our preclinical and engineering teams are like working in unison, it's humming, right? You're getting data out. The engineers and the comp sci's are analyzing it overnight, feeding back into the next day's experiments. Like that's not really possible clinical studies. There's this barrier between you and the hospital, where you're like waiting for your data, and then you have to wait. Then you have to submit new ethics to like run a new study, etc. Being able to turn that wheel super fast would be pretty exciting.
Sorry, this is leading into a lot more questions, but I am just now realizing I'd never actually asked what the experimental loop of well, actually, is there an experimental loop that goes on
in rodents?
At Coherence, like where like you design like one version of the device, you implant it, you see how well it works, or is it kind of like you have the device, it's the development is kind of finished, and you're now just seeing
okay, constant iteration. Both on our preclinical, all of our preclinical devices, where we're recording data from these animals, running new stimulation regimes, and on the sort of primary product development pathway as well. Both of those we have iterative, like tight iterative loops.
You exist amongst many other neurotech companies, and you're probably the most alien amongst many of those neurotech companies. How do you do you pay attention to most of the neurotech research that's going on outside of your immediate field, or it's not super applicable to what you are doing?
No, I mean, firstly, I take it as a great compliment to be called the most alien neurotechnology company. We're fine. That's good. Thank you. So secondly, no, I mean, both of us certainly are having conversations almost every day about what's going on in the field. It's entirely relevant, both from a technology landscaping exercise, but also from a cultural landscaping exercise. Like, which indications are getting more heat in the use of neurotechnology? Where are people most excited? What are the innovations that are happening that are convincing more and more clinicians and patients to adopt these technologies? Like, we need to be abreast of all of this, because there are some similarities with the technology stack and how it's introduced to the patient as well.
Do you think you're a neurotech company with ambitions to attack cancer, or a cancer company with ambitions to use neurotech?
I think, I personally, I'm a neurotechnologist that wants to develop technologies that can help a lot of people, and oncology seemed like the fastest and highest impact route to get there. If I can speak on behalf of Elise, and maybe she'll say I'm wrong, I think Elise comes more from oncology matters, and I'm going to use whatever tool I can to help these patients, and this kind of makes sense. Is that an accurate read on you?
Sure, I think so.
Oh, you disagree?
I think so. I don't know why the two have to be
Sure.
I don't know why they have to be separate. I feel like I think that there are so many debilitating conditions and diseases that we could focus on and that need attention, and there's like two major diseases in the world that are causing death or suffering for a lot of people, and that's like cardiovascular disease and cancer. And I feel like we can have a really big impact here by leveraging technology that is well established in other indications that could have huge potential in cancer. So I don't feel like I fit in either camp. I think I want to develop technology that will benefit people.
That's fair. I'm more neurotechnology pilled. I'm like, neurotechnology is crazy. Why are we not using it in all these indications? It's amazing. It's going to change everything. To the extreme cases that some of the neurotechnology and BCR companies are making, to just like day-to-day medicine.
Yeah.
I just think that cancer is an extremely promising way to get there and to scale these technologies into a lot of people.
Do you suspect that the full landscape of possible perturbations is pretty limited and you've like discovered most of them, or you may actually expand that over time?
I would say initially it's like looking at well-established, regimes. So, if you were to take what Setpoint or Galvani were doing in vagus nerve stimulation for rheumatoid arthritis, they're targeting immune response there. Well-established parameter sets that are published in literature and have been done in human, like those are the kind of more safe bets that you'd want to try in a novel indication. So I would we are starting with those types of things with some variations that depend on the nerve that you're targeting, whether you want to again modulate, so increase immune function or immune activity, or decrease stress function. They're very different types of stimulus that you'd apply, but they are well established.
It's actually a real problem in clinical programming generally, not in our field, but in other fields, for example, pain, where the clinical programming profession hasn't caught up with the engineers. So you've got more and more complex devices. You've now got hundreds, in some cases, of electrodes with thousands of different potential waveform characteristics that you could apply to each electrode, which gives you this multi-billion parameter operational space. And then you've got a clinical programming nurse sat there saying, where do I even start on this? Mm-hmm. There's this massive space now that I have to operate in to try and treat the pain of this person. And it's a job that probably will end up being done by some AI model down the road. Mm-hmm. I'm using some sort of Bayesian optimization, not a nurse going, does it feel better or worse by doing this?
Is that currently how it's done?
It's currently how it's done.
Okay.
I think you'd be quite surprised how much human in the loop there is in electrophysiological medicine, where you've got people watching a screen saying, I think they're going to have a seizure soon. That's something that we've actually seen. And someone else going, oh, well, better douse their brain to stop that happening. And there's no model, there's no computer really in the loop giving early indication.
You mentioned a little bit about what gives you anxiety, Ben. I'm curious what gives you anxiety, Elise, or if you'd like to add to your answer.
I think for me, it's I would say it's anxiety. It's a combination of anxiety and frustration. It's you want to move as quickly as humanly possible. The impact that we need needs to be done in humans, and you need to get to humans as quickly as possible. But you don't have all the answers in that design process, and that is frustrating and can be maybe anxiety inducing. So you're having to make some assumptions about what might happen in certain scenarios, or how to design this implant, and it has to be safe, of course. So that's probably that iteration. You just want to get to humans as quickly as possible, but you have all of these things that you need to consider, and that's frustrating. Drives me a little insane.
Yeah, I mean, I totally agree with that. I mean, you work in cancer field yourself, correct?
Mm-hmm.
And we're not in the ads business. We're not interested in pumping out a few extra targeted ads to people. We're in the game of actual human beings who are dying quickly, and we're trying to get technologies that can help those patients as quick as possible. That is frustrating. That is anxiety inducing, especially when you maintain a close connection with those patients and you see those patients dying, and then you're screaming at people in the office to move quicker because you're very connected to that.
I don't think I have any other questions. This has been an amazing conversation. Thank you so much, Elise and Ben, for coming on.
Thank you so much. It's been a pleasure. Have fun.